Thanks very much for inviting me to speak today. This is work done by myself and my two graduate students, Andrew Harrington and Benjamin Thomas at the University of Georgia. So this is work that's part of a collaboration that came out of the IBIV meeting 2 years ago. So the name of the project is QSTORM, Switchable Quantum Dots and Adaptive Optics for Super Resolution Imaging. And so we're one of 5 groups that are involved in this research, and you're going to be hearing from Gang Ron from Jessica's lab at Ohio State University, and then Beth from Brown University, Guy from Carnegie-Mellon, and finally Carol Lynn Alpert from the Boston Museum of Science, who are going to be talking about aspects of the project. So the idea, I think, is sort of self-explanatory from the title of the project. We want to add sort of 2 components to localization-based microscopy. We want to use quantum dots and adaptive optics along with that technology. So localization-based microscopy is a technique that was published by 3 different groups sort of roughly simultaneously in 2006. And it goes by sort of many different acronyms. So STORM came out of Xiaowei Zhang's group at Harvard. It was called PALM by Eric Betzig at Genelia Farm, and FPALM by Sam Hess at University of Maine. They basically describe the same technique. There's just been a sort of a plethora of acronyms that sort of describe the different sort of techniques, and mostly the acronyms have to do with sort of different labeling strategies. So we're going to use the acronym STORM, which stands for Stochastic Optical Reconstruction Microscopy. And the idea behind the technique basically is that in a wide field image, if you image a point source, you're going to get sort of a blob of light with a width that's given roughly by the wavelength of the light divided by 2 times the numerical aperture. And then for a high resolution microscope, this is sort of comes out to roughly 250 nanometers. So if you image a bunch of fluorophores that are close together, this is sort of what you're going to see in a typical microscope. You're just going to see one spot of light with the width that's given by, again, roughly the width of the airy disk of light, which is caused by the diffraction in the microscope. So let's say you have a ring of fluorophores like this, sort of with a radius of 100 nanometers. You're going to see something like this. If you were just imaging one fluorophore, you can figure out where the center of the emission is much more accurately than the width of the emission spot. So basically you can think of this, if you're climbing a mountain, you can figure out where the top of the mountain is pretty accurately. You're not limited by how wide the mountain is, right? So it's sort of the same idea. And so if you're just imaging one fluorophore, then how accurately you can figure out where the center is basically determined by photon statistics. So the more photons you collect, sort of the more accurate a picture you have of this spot, the better you can figure out where the center is. So you could imagine that if you could sequentially image each of these fluorophores and figure out where the center it is, you could plot out that center, image the next one, plot its center, and then you could end up with an image like this instead of an image like this. So instead of your resolution being limited to about 250 nanometers, you'd be limited to something, so typically maybe 20 to 30 nanometers instead, and you get a much better resolution image. And this is just sort of the mathematics of what the statistics looks like. So basically it's sort of the same calculation as calculating the error of the mean, if you want to think of it that way. So to make this technique work, what you need to be able to do is you need to be able to look at each individual fluorophore at a time. So you need fluorophores that are photo-switchable. And to get good statistics, so to be able to localize each fluorophore accurately, you need to have a large number of photons emitted from each fluorophore. So sort of the current technology, sort of the best sort of numbers people typically get are maybe 5,000 photons from each fluorophore before it irreversibly photobleaches. And the switching time, so you can have sort of duty cycles where each fluorophore is on maybe for 1/1000th of the time, so sort of a switching ratio of 1,000. And so to maximize performance, this type of imaging is usually done on very thin samples. And to make a thin sample, a lot of the techniques were at least initially done with total internal reflectance microscopy. So in this technique, you're basically illuminating only about a few hundred nanometers just below the cover slip of your microscope slide. And then more recently, people started using sort of oblique angles of illumination. This is also called high-low microscopy. And here you're illuminating maybe 1 or 2 microns below the cover slip. And so this is just another sort of cartoon explaining this technique. So basically, if you have a structure that's a ring and you label it with a bunch of fluorophores, you want to be able to image each fluorophore at a time. So if this fluorophore is on, you're going to get a blob of light like this, which is pretty big compared to the structure. But if you can localize that center and plot it, and then image the next one, you can sort of map out your structure. And for this to work then, let's say you have 25 fluorophores attached to this ring, you basically only want to have one fluorophore on at a time, which means that you're going to need a duty cycle here of 1/25 to make sure that on average only one fluorophore will be emitting light at a time. So there's sort of two aspects of resolution in STORM microscopy. One, as I mentioned, is the photon statistics. So the more photons you collect, the better your image is of the point spread function, or the image of one fluorophore. And the better that image is, the easier it is to tell where the center of emission is, and the better your localization accuracy is going to be. And the other aspect of the resolution has to do with how well labeled your structure is. And if your labeling is fairly sparse, you're not going to really be able to tell what the structure is. Right? You just don't have enough information. Even if you can localize each of these fluorophores very accurately based on the photon statistics, if you don't have enough fluorophores, you can't really tell what's going on. As you increase the labeling density, so as the density of your fluorophores in your sample gets bigger, you get a much better idea of what the structure is. And so this sort of mathematical idea behind this is known as Nyquist sampling. And basically the idea is if you want to be able to get spatial frequencies, say of 50 nanometers, your labeling would need to be 25 nanometers. You need to have labels 25 nanometers apart to get that kind of accuracy. So the higher the resolution, the higher labeling densities you need. So if you want to do this in thicker samples, you encounter a couple different problems. So one is that if you start imaging through a thick sample, your point spread function, so the image of one single fluorophore, is going to get degraded. So you're going to go from some nice cone of light with a high peak intensity to something that's got a much lower intensity and sort of a distorted shape. And this is due to aberrations which both reduce the peak intensity and distort the shape. And that distorted shape of the PSF means it's going to be harder to get a good localization of the emission. You're going to have worse photon statistics, and you're going to be looking at a larger structure to begin with. And the other problem is that you're going to have sort of background light from other fluorophores in your sample if you're imaging through a thick sample. And so that means that when you look at your photon statistics, so here we're plotting the resolution you get versus the number of photons. If you have no background, you're on this black line, and with 10,000 photons, you can get sub 10 nanometer accuracy in the localization of PSF. As you increase the noise from background photons, so if you have sort of a fluctuation of 100 photons due from background from other fluorophores that are out of focus in your sample, then that curve degrades rapidly. So then with 10,000 photons, you're only getting maybe 60 to 80 nanometer resolution. And as the background noise gets worse, that resolution gets worse. So if you want to get high resolution when you have background light, you've got to need to up the number of photons from each fluorophore to get better photon statistics. So here's just some plots showing. So if you have as you're illuminating a thicker slice of your sample, your resolution is going to degrade. So here the high resolution is the darker blue. And you're going to need more photons to get high resolution. And this is also going to depend on this blinking ratio, so the fraction of fluorophores that are emitting at a time. So now those not only can contribute to your in-focus ability to localize one fluorophore at a time, but it's also going to contribute to your background noise. So if you have a lower ratio, so if you have more fluorophores on at a time, you're going to have more background light. So if you want to get high resolution in thick samples, you want to have more photons emitted so you can get higher resolution even with background, and you want to have a lower ratio. So you want to be able to say, okay, I only want 1 in 10,000 or 1 in 100,000 of my fluorophores emitting at a time, so you have less background light and you can get higher resolution. So that's sort of the challenge that we're trying to address with this project. So in thicker samples, you're going to have background light and you're going to want more photons to get you a better localization. And you have to worry about the thick sample itself distorting the image of each individual fluorophore. So our goal in this project is to use two technologies to allow STORM imaging in thicker samples. So we're going to use quantum dots to achieve better localization. So quantum dots produce more photons for a given amount of time and a given intensity than other fluorophores, than small molecule fluorophores and fluorescent proteins, and that allows higher localization. And also they photobleach significantly slower, which means that you can image them for longer and get more photons that way. And then we also want to use a technology called adaptive optics, which corrects for aberrations so that we can have a nice, highly corrected PSF with a high peak intensity and a small width, even thick into the sample where we're looking at where we have to deal with aberrations. And also, since we're using adaptive optics, we can use that to introduce astigmatism into the PSF, which allows you to do three-dimensional STORM imaging. So this is sort of just to give you an idea of sort of what kind of resolution you can get in STORM. So this is the image of a fluorescent bead on the microscope, and each pixel here is about 90 nanometers. And we just, to see how stable our microscope was, we imaged this bead for about 40 minutes. And this is sort of what, and we localized the center of emission for, every minute or so over 40 minutes. And this is the curve we got. And you can see that there's a sort of small amount of movement of the microscope stage over that time frame, sort of in a diagonal direction. But that movement is less than 100 nanometers. So it's barely more than the size of one pixel. So this PSF is barely going to move in that amount of time. But again, you can see that there's a lot of things that are happening that you can discern if you do these localization techniques. You need photo-switchable fluorophores for STORM, so we need to develop sort of photo-switchable techniques for turning off and on quantum dots if we're going to make them work with STORM imaging. And one of the techniques that's been published in the literature is called quantum dot bluing. And the basic idea here is that if quantum dots are in the presence of oxygen, they're going to slowly oxidize over time. And as they oxidize, the core diameter of the quantum dot is going to get smaller, which means that the quantum confinement is going to increase and the emission wavelength will increase. So as the quantum dot oxidizes, its emission wavelength is going to move to the blue. So if you look at an emission window, a wavelength window that's to the blue side of the normal emission wavelength of the quantum dots, as they oxidize, they're going to move into this emission window that you're measuring, and they'll emit light. And then as they oxidize more, they'll move out of there, and you won't be able to detect them anymore. So since the oxidation process is stochastic, so they oxidize at different rates, you're going to see a small fraction of your quantum dots moving into this wavelength window that you're imaging, where you can look at them, and then they'll move out of there, and then you can localize some more. So as they move into this blue window, you localize the center of that particular quantum dot, and then it leaves, another one moves in, and you localize that one. So this is one method for using quantum dots in STORM imaging. And here's just an example of a bunch of quantum dots on the cover slip. So you can see them sort of blinking as they move into this window of wavelengths. So these are 545 nanometer quantum dots, and we're looking at them with an emission filter that's right at, I think, 512 nanometers with a 10 nanometer bandwidth. So initially we don't see them, and as they oxidize, they move in, and they flash on briefly before they move out of the window again. And if you localize each of these guys as it blinks on, you can sort of build up a high resolution structure. Again, this is just quantum dots sitting on the cover slip, but you can start to see all the structure with about a 30 nanometer resolution here of where they all are sitting. So another technique we've been exploring is using magnesium doped zinc selenide quantum dots. And then these quantum dots normally emit in the yellow right around 560. And so you excite them with a blue light, and then the excited electron will fall down to a trap state from the magnesium, and it'll fluoresce going from the excited to the ground state of the trap. If you, in addition to the blue laser, which you're using to excite the fluorescence, shine a red laser onto your sample, you'll be taking the excited electrons out of this trap state and re-exciting them, and they're going to lose energy through a non-radiative mechanism, which means that if you turn on the red laser, you're going to reduce the emission intensity. So this is another way of modulating the intensity of a quantum dot. So here's just an example. If you don't have the red laser on, you get a bright spot. If you turn the laser on, this intensity diminishes, and you can look at the modulation intensity here. And as you increase the intensity of your red laser, you can increase the modulation depth. And in theory, you should be able to get close to 100% modulation. Another technique, which Gang is going to talk more about, is come out of this project, is to encapsulate the quantum dots in micelles to improve their stability. While Gang and Jessica are developing these photo-switchable quantum dots, we've also done some imaging using Alexa dyes. So it's been shown by Marcus Zauer's group that you can use conventional dyes for STORM imaging, and that the Alexa dyes have been shown to have good properties. And the basic way this mechanism works is that a fluorescent dye, typically you excite the electron to the singlet state over here, and it falls back to the ground state by emitting a photon. That's the normal fluorescence mechanism, and that happens with a lifetime of on the order of 10 nanoseconds. There's a small probability each time it's excited that it's going to go over to a triplet state where it doesn't fluoresce. And from that triplet state, the molecule will get reduced, so it'll add an electron, and it'll just sort of sit in this non-radiative state. And it can sit there for a very long time. So if you keep on exciting all the fluorophores in your sample, you're going to have a situation where a lot of them over time, a bunch of them are going to move to this triplet state, and they're going to sit there, and they can sit there for several seconds. So you can have a situation where you've moved. So if you excite your sample with high fluorescence for a time, you're going to have a situation where you've moved most of these fluorophores into the triplet state where they're not fluorescing and only a small fraction are still radiating. So the way you can use this for STORM is that you'll move most of your fluorophores into a state where they're not radiating. You'll have a small fraction of them that are still emitting fluorophores, so you can localize those. And then over time, those guys will move to the triplet state. Some other ones will eventually come back from this non-radiative state back to the ground state where they can now fluoresce again, and you can localize those. So with this technique, you're going to have a small fraction of them that you can use for localization in the fluorescent state and a much larger fraction in the non-fluorescent state. And so that works because this radiative decay from the triplet state is much slower than the rate in which they move into the triplet state. And this determines sort of the blinking ratio or the fraction which are on at a time. And so typical values are about 1 in 1,000 that people have achieved with this technique. And again, you can get about sort of 5,000 photons out of an Alexa dye before it photo bleaches. So let me just show you some examples of the imaging that we've done. So this is from Beth's lab. This is myosin heads labeled with Alexa 48 and rabbus psoas muscle. And this is just sort of the raw data. You can see individual fluorophores turning on over here over time. And as each one gets turned on, you localize its center of emission, and you plot it over here. And if you take sort of 10 to 20,000 images, you can build up a high-resolution STORM image of the sample. And so you can see some of the structure here. We're not really in a position to talk about the biology yet, these sort of early images, but we're sort of happy with the way the imaging is working. We're getting sort of good localization of individual molecules, and we're building up sort of a high-resolution image of the muscle. So another example is from Guy's lab is looking at tau protein in PC12 cells. So this is a neuronal process coming from the cell body. And again, here's the raw image where you're looking at individual Alexa 488 molecules that are blinking on and off. And then you localize each one as it turns on, and you can build up a high resolution of where the tau protein is along the process. So we've done some comparisons of Alexa dyes and quantum dots using the bluing process that I talked about before. And we are seeing more photons coming out of the quantum dots as compared to the Alexa dye. So here you have an average of, from this experiment, we have an average of 1,500 photons from the quantum dots compared to 1,100 for the Alexa dye. So these are histograms of the number of photons emitted by a molecule before it photo bleaches over thousands of molecules. So you get more photons from the quantum dots, and you also get the maximum. The tail extends much higher, so the maximum here we counted was 30,000 photons from one quantum dot as compared to 17,000 for the Alexa dye. And with the quantum dot technique, you can also crank up the intensity to get more photons. Since the bluing technique is not, the amount of time they spend in the window where they emit photons is not directly related to the intensity that you're using. So you have some control over the number of photons that way. Okay, so let me just wrap up quickly. So far the images I've shown have been mostly in thin samples, so maybe 1 micron thick or so. Here we've also done some imaging sort of in zebrafish embryos, and we've been able to sort of show that using Alexa dye you can look at the blinking of individual dots here. And again, you can see there's a lot of background, and the spots aren't as sharp as they are for thin samples, which is sort of why we're working on quantum dots and adaptive optics for this technique. So I haven't really talked about the adaptive optics at all, so I'm just going to talk about that very briefly. There's going to be more. You can look at our poster later that Andrew will be showing, I think, tonight. So astronomers use adaptive optics to correct for optical aberrations in the atmosphere. If you look at, this is Neptune. If you look at it just with a ground-based telescope, you see a big blurry spot because the effects that make stars twinkle also make everything get blurry. If you use adaptive optics correct for those to turbulence in the atmosphere, you can get a much sharper image. So we want to apply that technology to microscopy to get sharp images of each fluorophore as it blinks in a thick sample. So here's just a quick example of what this is a C. elegans worm, and if you look at some beads that are on a cover slip below the worm, you can see that they can be strongly distorted by the body of the worm. And you can use adaptive optics, which is a mirror where you can locally deform the shape to get much sharper images from each of these spots by correcting the aberrations. So, and again, there's going to be more of this in our poster. So I think I'll just wrap up. There's sort of some other work done on using STORM in thick samples using selective plane illumination microscopy and two-photon activation. And so these are sort of complementary techniques that we can combine with the quantum dot approach that we're taking. So in conclusion, we've demonstrated that you can do STORM imaging. And we've looked at thin samples with Alexa 488 labeling. We've started to use quantum dots for STORM imaging, and the next step there is to integrate them into biological samples and to use the photo-switchable quantum dots that are coming out of Jessica's lab at Ohio State. And we sort of demonstrated some adaptive optics, but we need to do sort of more work combining that with the STORM imaging. And thank you very much.